Hello everyone and welcome to Infotainment Lounge. This live stream is for A2 level chemistry students and this is about the DNA replication. We studied the structure of DNA in the previous video. Today we are going to deal with the DNA replication. I know the non-biology students had a lot of problem in the previous live stream, so I will try to keep my pace slow, but at the same time I want you all to be very attentive. If you are a non-biology student especially, I want you to be very attentive because this stuff is new to you. Okay, now let's start with the DNA replication. Now, before moving on to the DNA replication, I want you to give some basic information about living organisms, especially to non-biology students. See, if you are a non-biology student, you must know a few things first. See, the bodies of all the living organisms are made up of small units known as cells. It doesn't matter what type of living organism is that, the smallest segment of the smallest block of the living body is a cell. If you are a human being, if you are any other animal or a plant, if you are all the living organisms, even the microorganisms, the smallest part of a living organism is a cell. Okay, now let us take the example of humans. A body of an adult human is made up of many cells. How many cells? Billions of cells. There are a lot of cells. And almost all the cells contain DNA. DNA is the genetic code for living organisms for the production of their proteins. So almost all the cells, not all the cells, biology students would understand why I'm using the term almost, but you don't have to go into that. For you, almost all the cells have DNA, and almost all the cells, they all have those cells who have DNA, they have exactly same DNA. Like if you remove one cell from your hair and you remove another cell from the fingertip of your foot, both of those cells will have exactly same DNA. That is why a similar set of proteins can be produced all around your body in different cells. But originally, when a human being was not an adult, he was an infant, he was a child, then he was, before that he was not even born. Initially, when a new living organism, a new human being is formed, its body starts from one cell. Then those cells keep on dividing. One cell divides to form two cells, then two cells divide to form four cells, and similarly they keep on dividing during the growth period up to billions of cells. So the first cell is called a zygote. The first cell, it had a specific type of DNA, specific sequences of bases in the DNA. And after adulthood, all the billions of cells in your body, they all contain the same DNA, the same sequence of amino acids which the original cell had when it was first formed, the zygote. So that means there are copies of the DNA being formed during those cell divisions, and those copies are distributed among all the cells. This is why the DNA combination, the sequence of DNA does not change throughout the life. So those copies have to be exact copies, like exact photocopies of the original DNA sequence. How are they formed? Those copies are formed during the process known as DNA replication. Please let me know in the comment section if you have understood this much about the living organism's body. Then I will move on with DNA replication. Okay, you want me to give you a recap. Okay, I'm repeating it again. See, all the living organisms, all the bodies of all the living organisms are made up of tiny units. Those units are called cells. Cells contain DNA. The DNA coding, the sequences of DNA is specific for every organism. Like your sequence of DNA is different from mine. Mine is different from anybody else's. Okay, but the DNA combination in all the cells of your body is the same. The DNA combination, the sequences of nucleotides in your DNA is different from the sequence of nucleotides in my DNA. But the sequence of amino acids in your DNA from one cell is exactly the same if you take out the DNA from another cell of your body. That is exactly the same. So basically, originally the body started from one cell. The body started from one cell. That cell kept on dividing. One cell divided into two, then two cells divided into four, and they kept on dividing up to billions of cells. So that is how you have billions of cells. But all those billions of cells have same set of DNA in your body. That means during that cell division, there are copies of the DNA being formed. The original DNA combination does not change unless there is an abnormality. Under normal circumstances, that DNA sequence does not change in any living organism's body. And why is that? That is because of copies of the DNA being formed. And that process during which the copies of DNA is formed is called DNA replication. So during DNA replication, exact copies of DNA, the original DNA, are formed. So now let us move on with the DNA replication. Okay, now DNA replication takes place in several different steps. First of all, the double helix of DNA starts unzipping. Starts unzipping. What do we mean by unzipping? See, this was the double helix, the two strands of DNA. They had sequence of nucleotides. If we know the sequence of nucleotides on one DNA strand, the other we can predict, because I told you the complementary base pairing thing, because they have hydrogen bonds between them. Suppose there is adenine here, this is an imaginary sequence. But the sequence on the other strand would be according to the first strand. And these were held together by hydrogen bonds. I told you there were 2 hydrogen bonds between adenine and thymine, whereas 3 hydrogen bonds between cytosine and guanine. Between all of them. So these are hydrogen bonds. So these hydrogen bonds can be considered as a zip. See, there are 2 separate strands which are attached to each other because of hydrogen bonds between the nucleotides, the bases of the nucleotides which are facing each other. So this is the force of attraction which is holding the 2 strands together. So this force of attraction starts breaking up from one end and continues breaking up to the other end. That is called the unzipping of DNA helix. See, these hydrogen bonds start breaking one by one by the action of an enzyme known as DNA helicase. It starts breaking the hydrogen bonds between the nitrogenous bases of the nucleotides of the opposite complementary strands. So this process is called unzipping. Okay, so when this keeps on breaking down, both the strands start detaching from each other like this. See, from this part they are still bonded to each other, but from this part they are detached from each other. And that action took place because of the activity of the enzyme DNA helicase. It broke off the hydrogen bonds between the complementary bases. Now, these are separate. Suppose I am taking the same sequence again. This is A, this is T, C, G, T, T, G, B, C. Okay, now there are free nucleotides present inside the cell already. Where did they come from? How they were formed? Non-biology students do not need to know that. But you just need to know that free nucleotides are already present in the cell. They start arranging themselves in front of these nucleotides. And again, the sequence would be specific because there is always a complementary base pairing between the nitrogenous bases. So new nucleotide with thymine will place itself in front of this adenine nucleotide. A new nucleotide with guanine will place in front of this cytosine. Similarly, it would carry on. These are the nucleotide, and how and why are they being arranged in front of each other? That is because of the hydrogen bonding. They start forming hydrogen bonds with each other. Three hydrogen bonds between cytosine and guanine, and two hydrogen bonds between adenine and thymine. Similarly, complementary bases of these nucleotides would start arranging in front of these bases. Like in front of this thymine, there would be an adenine. It would form hydrogen bonds with this one. Then in front of this guanine, there will be a cytosine. It will form three hydrogen bonds with this. Then a guanine here, three hydrogen bonds, and so on. See, these nucleotides are not bonded to each other so far. They are separate from each other. But they start forming hydrogen bonds. Hydrogen bonds are being formed in the next step. And this reaction, the formation of hydrogen bonds between these complementary bases is controlled by another enzyme. It is known as DNA ligase. Okay, I have a couple of questions. One is that are the details of mitosis in our syllabus? Answer is no, it is not in your chemistry syllabus. See, you are dealing with the chemistry of these biological molecules and how this works. The whole mitosis thing and meiosis thing, that is not a part of chemistry syllabus. That is a part of biology syllabus though. Mitosis is a part of AS biology, meiosis is a part of A2 biology, but not for chemistry. Okay, then a student is still confused about DNA helicase. Okay, no problem. Keep on asking me if you ever get confused. Just let me know. I will explain the things again. I will try to go slow. Okay, DNA helicase segment. You said you did not understand the DNA helicase segment. DNA helicase is just an enzyme. And biology students are familiar with these terms which end with ASE, which end with A's. All the terms related to biology which end with ASE, they are all enzymes. And enzymes, what are enzymes? Even if you are a non-biology student, you already know that enzymes are biological catalysts. They speed up a chemical reaction without themselves being chemically changed. So enzymes carry out specific reactions. They speed up specific reactions in the living organisms' bodies. So what type of reaction is sped up by DNA helicase? It is the breaking down of the hydrogen bonds between complementary bases. Like these two hydrogen bonds between this adenine and this thymine can be broken up in the presence of DNA helicase enzyme. So during DNA replication, initially DNA helicase starts acting. It starts breaking up the hydrogen bonds between the complementary bases, due to which the strands of DNA starts getting separated from each other. This process is called unzipping of DNA. Got it till here? Moving on to the next point, the next type of enzyme, it is DNA ligase. Again, it is an enzyme. That means it has to speed up or control a specific type of reaction. Which type of reaction does it control? Opposite to that of DNA helicase's reaction. DNA helicase was breaking hydrogen bonds between nucleotides. DNA ligase forms hydrogen bonds between the bases of complementary nucleotides. But this time, these hydrogen bonds are not formed between the two already existing strands. In fact, these new hydrogen bonds are formed between the old strands and new free nucleotides. That reaction is controlled and sped up by the enzyme known as DNA ligase. So when this keeps on unzipping, the original two strands would be separate from each other completely by the end, and there would be, suppose this is the original, this one, the strand on the left side, so it will have the same sequence obviously. And this is this strand, that means it has the sequence that it had before. Nothing is changing in them. They just broke off from each other. Their nucleotides are still bonded to each other in the same sequence. And now the new nucleotides have arranged themselves in front of these. This sequence took place, this process took place because of the enzyme DNA ligase. and these nucleotides would always be complementary to those which are already there. And there are hydrogen bonds between them. These hydrogen bonds were formed because of DNA ligase. Okay, but these nucleotides, these new nucleotides are not bonded to each other so far. And that bonding between these nucleotides is the last and final step of this DNA replication process. And that is controlled by another enzyme known as DNA polymerase. See, I told you earlier that nucleotides are the monomers and DNA strands are the polymers. So, DNA polymerase is another enzyme which carries out this polymerization reaction between the nucleotides. It starts forming the phosphodiester bonds between these free nucleotides. These are not intermolecular forces, these are full-fledged bonds, covalent bonds, and these linkages are called phosphodiester bonds. So that means when this polymerization process keeps on taking place, a new strand of DNA is formed. This is the old strand which came from the original DNA. Again, this one is a newly formed DNA strand and this one came from the original DNA molecule. See, but now there are two sets of DNA double helices after this. First, let me note this down. So two new double helices are formed because of two new DNA strands being formed, but each of the double helix, see, consider this one double helix. This double helix contains one of the old strands and one of the new strands. Again, this double helix, it also contains one of the new strands and one of the old strands. But if you look at the sequence of nucleotides in both the strands of each double helix, that sequence is not only same in these two, but that is exactly the same as the original one. I'll give you a few seconds to take a look at that. See, this is how new strands of DNA are formed and new sets of DNA are formed which are exactly similar to the original ones. Now, what if cell division is taking place? If there was one cell, it needed one set of DNA. That cell is now dividing into two cells. Both of them needs a set of DNA each. Now, during DNA replication, the new sets of DNA are formed, which are exact copies of the original DNA. So one of the new cells will take one set of DNA, and the other new cell being formed will take the other set of DNA. This is how, when the cells keep on dividing, our body started with one cell. Now we have billions of cells in our body. All of those cells originated from one single combination of DNA, and today in all the billions of cells in the body have that same combination of DNA. This is because of this process, DNA replication. This is how it is formed. And you have to remember another thing, that when new strands are formed, the new DNA double helices are formed, one of the strands is the original one and one of the strands is the new one. And this type of DNA replication is called semi-conservative replication. If you are a non-biology student, you don't have to go into the other types of replication like conservative and dispersive replications. There are other types, but you don't have to go into them. But you just need to remember this term, that when new molecules of DNA are formed, they contain one old strand and one new strand. Each of the double helix contains one old strand and one new strand, and this type of replication is called semi-conservative replication. Okay guys, now I want you all to complete this first, complete your notes, then revise, then read out all the content that you have studied so far, and then let me know if you did not understand any of the part or if you have any questions about it. I will not go into the next topic in today's class. I want you to stick to this topic so that you don't get confused. We'll start with the applications in the next class. So I want you all to note this down complete first, and then read it again, revise the concept in your mind, and ask me if you have any questions. Yes, the proper biology segment is just this much in your syllabus. Now we'll start with the applications. We'll start with electrophoresis, and that part is somewhat related to biology a little bit, but not that is a chemistry technique actually. The proper pure biology segment was up to this mark, and non-biology students must consider yourselves lucky, because up till 2017 A2 level chemistry syllabus included a lot more of biology syllabus in this segment, much more than it is now. Much of that syllabus has been eliminated from this A2 level chemistry, so you consider yourselves lucky. We have a question, but let me see who is asking if you are a biology student or not. Okay, you are not a biology student. Muhammad bin Imran, you are not a biology student, but so you don't need to go into that where are the new free nucleotides coming from. You don't need to know that. So I will not go into that. Okay, I have another interesting question. I'm seeing another very interesting question. It's again by the same student, Muhammad bin Imran, that are those DNA helicase, DNA ligase, and DNA polymerase, are all those enzymes already present in the living organism's body? I have a very interesting answer to this question. See, they are enzymes. And what are enzymes? I told you in the structure of proteins that tertiary structures, the globular proteins, they make up the most of the enzymes. So basically, enzymes are proteins in nature, and DNA codes for proteins. So that means there are specific genes in our DNA which codes for these enzymes, for the synthesis of these enzymes. So yes, they are already present in our bodies, but those are formed because of the coding present on the DNA. Because DNA codes for the synthesis of all the proteins. I hope I have not confused you even more. Yes, you're right. We are the cells which contain the DNA. Proteins are being manufactured inside the cells, and the cells contain the proteins, only those proteins whose genetic coding is present on the DNA. So yes, our cells are producing those proteins which are making up different things, like one of them is enzymes. Those enzymes are being formed inside our cells, and those cells are then carrying out many different reactions. One of them is DNA replication, which takes place before cell division. Okay, we have a question that why does unzipping of DNA takes place? But it has to take place, because if it doesn't, there would be no DNA replication. And if there is no DNA replication, then same sets of DNA would not be transferred to the new cells which are formed during cell division. So it has to take place, and in order to replicate the DNA, the presence of a few enzymes is compulsory, and those enzymes are already listed in this diagram and the descriptions. So those have to be present, and DNA has to be unzipped so that DNA replication may take place. Because if the hydrogen bonds do not break in the beginning, how are the new nucleotides going to arrange themselves in front of the old nucleotides? It would never take place. So the life would end with one cell. That is why it has to guys, if you have any more questions, I'm waiting for your questions. Okay, another interesting question from Mohammed bin Imran, who's a non-biology student, and he's asking some very deep biology questions. He might end up making interest in biology actually. The question is that does this replication carries out throughout the lives? See, there are some cells in which, like the cells which are involved in growth, like the cells involved in your muscles, in your bone marrow, in your skin, they are involved in growth. Yes, they keep on growth and even repair. They keep on dividing throughout your lifetime. And obviously when they keep on dividing, the DNA keeps on replicating for those cell divisions. Okay, but there are some type of cells which do not divide further after a specific age, such as the cells involved in your nervous system, like the neurons. Neurons are the cells which make up the nervous system of your body. They divide in the beginning, they keep on dividing up to an age when you become an adult, and after your growth starts when you are an adult, those cells do not divide throughout the rest of your life. That means they will have to keep the original DNA in them for the rest of the life to keep on producing the required proteins. But they have very strong mechanisms of repairing, because the cells can get physically damaged or chemically damaged due to several processes going on which are related to that. But they do not divide once you are a grown up, and they do not obviously the DNA in them does not replicate. Okay, yes. So the again the question that what happens when those cells of nervous system are damaged? Those cells keep on getting repaired. If the damage is beyond repair, then there are nervous system disorders. There are several different types of disorders in nervous system which could be which are possible actually, and because of different reasons, that may be a physical damage or that may be a chemical damage. But you don't need to go into that. You are going into too much of biology. This class is turning out into a biology class as I'm going further in that. So keep yourselves to this, and if you have any questions, regarding the structure of DNA or the replication of DNA. Just keep yourself to that if you are a non-biology student. And even if you are a biology student, I will do the biology details in a separate stream. I will let you know when I can do that. But so far, just for today, keep yourself to what we have studied today and in yesterday's class. If you have any questions regarding that, you can ask me. I'll wait for a few more seconds, then I will call it off. So guys, I think that is it for today. I'll see you in the next stream, and I will let you know when the next stream is going to take place. Most probably it won't be on the weekend. We'll do it on Monday. So wait till Monday for the next class. Keep on revising the previous work during these 2 days. Thank you, Allah Hafiz.